Okay, hello everyone, and welcome again to yet another journal club. So today it will be Zixian who will be presenting, so without further delay, the stage is yours, Zixian.
Hello everyone. We'll begin to discuss some questions.
Louder, maybe.
In total, there are 5 questions need to be discussed here, and I want to start with some easy ones. Firstly, it's have they seen a similar phenomenon also in other brain regions, neuronal types, in addition to cortex and SNCD neurons? The answer is no. They only checked it in SSP and SNC regions.
SSP again. So what is the SSP? Which that's a cortex, right?
Yes.
Which particular part? I just forgot. Just can you remind me?
This part.
Sensory somatic.
Okay, sensory somatic. Okay. And they didn't specify which particular neuronal group. Like they just did generally, right? So because my question was not only about the region but also about the neuron specificity. But like for this substantia nigra, right? So they looked for the dopaminergic ones, but for this cortical one, they didn't specify, right? Have they? Okay, it's under the synapsin promoter. I see that.
Exactly. Yeah.
Okay, I see that.
So definitely not GABAergic neurons, right? Because I think that synapsin one is not expressed in GABAergic neurons.
Yeah. That's also my understanding. Okay.
The second question is about the localization of the Pkm2 deletion. In this literature, Pkm2 was deleted conditionally. It's only in SIP and SNC. They used a Pkm2 flux mouse model and injected some specific AAV virus. Okay, and the third question is.
Excuse me. Can you get back to the second one? To this previous one. Okay. Yeah. Okay. Sorry.
Okay. And third question is, I want to see the behavior results when Pkm2 is knocked out in this story. They didn't check the behavior of Pkm2 conditional knockout. That is for the first part's answer, and the second part is about what will the metabolic parameters cognitive functions on motor ability changes when Pkm2 is knocked out. I want to start it with the motor activity firstly. I found some other literatures. They use some mouse model study and some behavior test found that when Pkm2 knockout, the mouse motor ability has improved.
Really?
Mm-hmm.
So in a Pkm2 knockout specifically in which cells is myeloid cells specific, and then they checked motor activity in the myeloid cells. How does it work?
About the stroke study.
Oh, okay.
So
okay, but that's well, that's a very special model, right? So like they're looking for motor activity after stroke model, right?
Yes.
Okay. but that's a different. Like here, we can talk about the somatic and the terminal localization of Pkm2 in myeloid cells, obviously, right? So that is a little bit different question here. Okay, but nothing else, right? Because I also had, like, it's a great question. I think it was Jakob who asked, because I think they show that there is a decline of dopaminergic neurons on the range of like 50%, something like that. I mean, this might still be compensated, but with some very sensitive techniques. You know probably which one I mean. One can actually detect even such declining, even if it is like around 50%. So I'm really surprised that they don't include anything related to the behavior in those mice which are losing dopaminergic neurons because of the deletion of this enzyme. So or maybe they did study that, but they couldn't find anything, and that's why they didn't include. That's also a possibility, right? Yeah. But I agree with Jakob that is a really good question to address, and that would be really like complete story, yeah, if they could also show that, yeah, indeed, so it can actually also impact the behavior.
You know, they because there's losing of 50% of the dopaminergic neurons in this model, it may be not shown in the phenotype, because in my previous institute there was some rodent analysis of Parkinson's disease, and you know they destroyed neurons with hydroxydopamine. And it was this behavior in animals, it's not so clear. So, you know, losing of only 50% of neurons may be not very efficient for modeling of Parkinsonism in rodents.
That is actually true, and that is exactly what I mentioned previously. So I mentioned that even 50%. So although it might not give the clear behavior phenotype, some studies claim that, okay, you might see some first signs when you lose around 70%. But what I mean is that there are techniques which are like extremely sensitive, so that you can maybe see some hints, but maybe they like, for example, if you use the accelerated rotor rod or like open field, you might not see anything. But if you use the constant speed rotor rod with some specific settings, so, and Xian is very familiar with that. So, and we also study the dopaminergic neurons. So that might show some hints, or at least some tendencies. Yeah. So, but yeah, so that's a good point, Jakob, and I agree. Yeah. So 50% could be not enough, indeed.
Yeah, I have a question, because I'm not this background. So, what is actually the myeloid cell? Is it like this type of stem cell?
Not necessarily stem cell. No, it's just this particular model, like this Cree. I don't know which exactly cell type does it cover, but this is just an illustration that in this stroke model, they also have this deletion of Pkm2, and they were studying for some motor activity, but in the stroke model, it's a little bit different. What actually Jakob was asking, so he was more asking about the dopaminergic cells. So this is actually not very relevant to this answer, but still interesting to see, like that somehow those cells, those are probably the immune cells, the myeloid cells, which are recruited to the site of the injury during the stroke. And that Pkm2 actually has to do with the activity, I guess. I don't know the story, but
it's like, say that the myeloid cells actually commonly can be found in bone marrow.
Yeah, not only, yeah. So myeloid cells are also like derived from the myeloid lineage. There are multiple different lineages of the hematopoietic cells. that are also recruited into the sites of injury. Yeah, but it's not the topic. It's kind of it's a little bit
like,
outside of the background.
Because I never heard it before.
Okay. Yeah, I think that's it, right? If we don't have any comments, Xian.
Okay. Also another research about Pkm2's function of motor ability. They use a Pkm2 inhibitor in rat with a diabetes model to study about what will happen when Pkm2 is inhibited. And in their results from the figure B2E, they found that when Pkm2 is inhibited, the motor ability is improved.
Oh, interesting.
Yeah.
Interesting. So, type 1 diabetes has, how do they link 6-hydroxy? I see they use 6-hydroxydopamine here to kill the dopaminergic neurons, as well as they use the streptozotocin to induce the type 1 diabetes, and this kind of compound effect is much stronger. This black bar in the latency to fall. Okay, and then if they use SK, is this chiconin, right? Is this okay? It significantly rescues. Is there a significance between this black and the last bar?
Yes.
There is. Wow. Okay, so this means that if the cells are really suffering, they're really struggling, those dopaminergic neurons, then the deletion of Pkm2 is not anymore beneficial. So I guess we can conclude that. So that it is. So since it might, I just speculate, here that probably those cells which are suffering, they would require energy somehow. And if you can produce only 2 molecules of ATP from 1 glucose, but not how many? 36. Then it might actually be detrimental for them, so that they cannot cope with this. And in that case, if you have such a strong challenge as streptozotocin and also this toxin, dopaminergic neuron-specific toxin, then inhibition of Pkm2 actually can be beneficial. Wow. That's a great paper, actually, what you found. That's very interesting. Maybe anyone has a different explanation, but that looks very interesting to me.
Okay, next is about cognitive functions. I found the literature published in Cell Metabolism. And they found that Pkm2 glycolysis is 4K12 IA Pkm2 positive feedback loop exaggerates microglial dysfunction, and when Pkm2 is knocked out, the cognitive behavior is improved.
Wait, so the knockout is where? In which? Only in microglia, right?
Yes.
Is it?
Yes.
So Pkm2 in microglia is detrimental. So
yes.
So you say that if you knock out it, then the cognitive function is improving, right?
Yes.
So Pkm2 is detrimental in the microglia, at least in the context of the cognitive functions, right? So which model did they use to exacerbate? Is it which did they use amyloid beta somehow linked? Like, what is the model? I see that they use the AD
Forgot.
And
I forgot.
they also studied the patients. Okay, that's interesting. So they claim that actually the glycolysis, but not oxidative phosphorylation in the microglia is detrimental, basically, right?
Yes.
If we kind of assume the simplistic way that Pkm2 down means kind of switch from the glycolysis towards the OXPHOS, right?
Mm.
That's very interesting. Okay. Cool. But a little bit different, right? Still, yeah. So like the previous one was exactly really fitting to what we studied here, right? So it was about the dopaminergic neurons and about this different localization of different function, right? So different way of producing energy in the terminals and in the soma. But of course, we cannot say anything like that about some small cells like microglia, which is also a myeloid cell, actually.
Yeah, I checked it. Okay.
Yeah.
Next is about the metabolic parameters change. It's the
just let that please.
Sorry.
I don't clearly know what to cover axon here. If you want to know some metabolic parameter, is it some TCA or mundane metabolism changes? Here is the literature about it. It shows that when Pkm2 lost, it can lead to some metabolic and protein expression changes. If you want to know something about, for example, the fat, and there is also literature shows that Pkm2 can function about brown fat adipogenesis.
Which is not a surprise, right? Because since it is such a critical regulator of the switch between how pyruvate is going to be metabolized, or whether it goes to the lactate or it goes to the TCA cycle, then we're not really surprised, right? So that it might change a lot, yeah? Okay.
Is it okay?
Are you satisfied? Do you have any comments?
Yeah, it's totally satisfied, and it was very complex answer. Thank you.
Welcome. The fourth question is.
Sorry. The same because that I already like more than 10 years don't study about human. So I wanna ask the last question 3.
Metabolic.
Yeah. I wanna ask about like the data, the color, what does it indicate? I only know like if let's say we're doing like the plants, right? So sort of there is like we do the microarray data.
Harry. So what they show here is that if you interfere with such a critical regulator of energy production as Pkm2, you get multiple different changes in many genes. And that's what they showed here, like multiple genes, and there is a change. You see blue is minimum on the bottom, and red is maximum. That's what it says. Some genes go up, go down. It doesn't say more. It just shows like there is this, like this is how you interpret this heat map. If there is a specific point they want to point to, so they usually like point it somehow, right? But this heat map says, okay, some genes go up, some genes go down. That's it.
Okay. And so this one is like use what technique to do that? If let's say plants,
it could be it's one of the omics techniques.
Oh,
So it could be sequencing. It seems that it's sequencing. You see, there's the volcano plot. It could be also a mass spec. It could be, so there are like any kind of like large scale technique can assess this kind of different genes, but you need to validate it anyway. Like it's just the beginning, it's just the first step.
okay, thank you.
Sorry for answering that. maybe you wanted to answer that.
The first question is, had this differential OXPHOS glycolysis balance been shown to be altered in some diseases or in aging? I found a literature about
Differential means, what I meant here is about terminals versus soma. This is what I meant. Sorry for not being specific, but this is actually what I meant. Differential means like that it is different in soma compared to the terminal. That is what I was referring to.
I didn't find the literature about
the
changes in soma.
It's my fault. I didn't specify it clearly. But okay, let's see what you have found.
I didn't find anything about this question.
Okay, but still, you have prepared, so we can go into that.
Well, Pkm2 studies, many researchers majored in cancer. They studied about Pkm2. That is associated based on TCA cycle glycolysis. And in the cancer-associated study, they found a lot about the OXPHOS and the glycolysis.
No wonder.
Yes. And so the literature published in Cell Metabolism shows that Pkm2 inhibited can rescue the triple negative breast cancer. And also a review found some checkpoint to see the how the cells use the OXPHOS and glycolysis, and what will happen at that time point. Also, another research shows that during brain development, different time point we use different the aerobic glycolysis ratio is different.
That's more towards the question, but still, it's like the initial question is a little bit different. So I was not specific enough. Okay.
And so the next question is about whether there are other genes or factors contribute to the differential activity in somata and the terminals. To be specific, I didn't find a very suitable literature. I just found some literature published in Cell Reports shows that the GLUT3 can function in glycolysis.
Yep, but here my question again is about this balance. What I mean, this differential is about this terminals versus somata. Only that. So I was pointing to this point. So had anybody seen that, for example, in some cells there is kind of more predominant use of mitochondria or the function of mitochondria maybe in the terminals than in the soma, which would be actually surprising for me, but still, like if they claim that could be also contribute, right? If you have more functional mitochondria there, so this might shift the balance of this TCA and OXPHOS towards the terminals additionally, like in addition to the Pkm2 function.
TCA, you said area means the Calvin cycle.
Yes.
So Calvin cycle.
The Krebs cycle. Right?
Yeah, the Krebs cycle,
not Calvin. Krebs cycle.
Okay.
So you couldn't see, yeah? So couldn't find it.
It mentioned the synapse and cell body.
Yes, indeed. They say because the proportional increase in energy expenditure with neural activity may be greater in the synapses than the cell body. Okay, here they say we don't know. Interesting. But they thought about that. But these guys addressed that, which is cool. Sorry, can you get back? There they say that GLUT3 deficiency compromise ATP levels at the nerve terminal, but not the cell body. Right, but if the cell body anyway does not actually produce much, at least in some of the cells that these guys addressed, that might be it is not so sensitive to the GLUT3 deficiency, since like ATP levels in the terminals, according to these guys again, is supposed to be higher, much higher. So like if you have some kind of challenge which decreases the like interferes with this pathway, this should impact the terminals actually more than the body. So it kind of aligns. It's again the speculation, but I think it aligns with what these guys showed, which is interesting.
Okay, next.
Next time. You wanted to say something?
No. Next is a review about the glucose metabolism in nerve terminals. It mentions that axons utilize a machinery similar to that in exercising muscle to regulate glycolysis. And one potential important but relatively poorly explored feature of glycolysis is that it can only proceed if there is a sufficient supply of the essential electron acceptor nicotinamide adenine dinucleotide. It mentioned that some other factors functioned in the balance.
That's interesting. This paper was from 2017. Okay. Interesting. Really interesting.
Next.
Yeah.
Also another two literature shows that it can function on the balance between glycolysis and OXPHOS, but I didn't look at it detailedly.
Right. Is it also about the differential? No, it's not, right? It's not about kind of like terminal, because my question was specifically about this differential function.
I didn't find something.
Okay, cool. Which tells that this research was really novel, right?
Yes.
I found it really interesting. Yeah, really interesting.
And that's all.
Okay.
So it means that the glycolysis also takes part, but there is no like specific data, because like normally that if I still can remember that, of course, that our brain neuron functions needs, like correlated with the glycolysis as well, and in our daily activity as well. So, is it like the question spot that there is no like specific the bigger the data?
No, it just shows that that this their data is novel. It's that it had not been extensively studied. That you have like increased activity of the oxidative phosphorylation in the terminals, and the increased function in the soma of the neurons. And this seems to be also to some extent beneficial to the neurons, unless they get diabetic or you start to treat them with some toxins, and then they actually suffer more. Yeah. So. Yeah,
I know that
a little bit louder, maybe.
I know that this Pkm2 is often overexpressed in cancer cells. So, and here they found that the division of this Pkm2 leads to oxidative damage and progressive loss of dopaminergic neurons. So the question is there like a link between this Pkm2 and the metabolic behavior and the cancer glucose, let's say? I mean, does this Pkm2 have non-metabolic function? I think it should have, but I just don't know about it. I just wondered.
Pkm2's function about the
Non-metabolic function.
metabolic,
Not non-metabolic.
not metabolic function.
Which can somehow contribute.
But in a way,
to
kind of, it's a matter of definition. In my opinion, like almost everything is metabolism. So in a way, right? So it's right, and this role in this function in the cancer, it very obviously, cancer also like usually also requires like more energy, right? So and that's why it is implicated in those cancer studies. So I wouldn't just separate that. I think it is really going aligned with. Mm-hmm. Also, Xian provided so many different links where there are tons of literature about the Pkm2 effects, including the cancer biology. So I'm not surprised at all. But for me, what is very interesting is this balance of these terminals versus soma. Yeah. So, and that is really like novel. I didn't know about that. It's really interesting. Anybody would like to comment or ask anything else? Since Xian knows, seems like everything on this field, so whatever you ask, she finds so many different examples. So great. So take the opportunity and ask her. So she is an expert, it seems. Okay. So if we have no further comments, Xian, maybe you can would like to wrap up and tell like what do you think about this study? What do you find it so well designed, well thought through, the methods adequate, or whatever, like everything what you want to say. So or vice versa, maybe you have maybe you found something hidden or something missing or whatever, or something lacking, something disturbing.
I found that literature just because it studied a little bit about the dopaminergic neurons. So I look at the I think it has a lot of work studying a novel thing. And the one thing impressed me is how they found the Pkm2 to study, because they, in one of their results, they have proteomic results. For me, if I studied about it, maybe I would choose some highly changed protein or genes to study. But they go back to the TCA cycle or that pathway and found the Pkm2 to study. That's a new insight for me to find some target gene to study. And that's all.
Wow, that's a great point. So you mean that actually based on the omics, on this proteome study, it seems that that's how they got to this Pkm2, right? And there were several hits that they were even higher than Pkm2, but they still...
They didn't mention that, but if Pkm2 is the top 10, I think they should figure in the hit map.
Mhm.
But they didn't point to that. Just in the figure, they show that Pkm2 expression is changed in two groups.
Mhm.
And then they study about Pkm2.
Oh, okay, and let me check it. So it's in which figure was that? Figure 3. So in the beginning, they only studied the... indeed. So in the beginning, they only studied the descriptive, right? So they just show, okay, there's something is wrong with the... Yeah. In the first two, I just forgot. Yeah, they only show that there is this differential function of these OXPHOS and glycolysis, and then that is what you mean, right? So they made this kind of it's proteomics. It's not sequencing. It's proteomics. That is proteomics. You see, so in this case, Harry, they use proteomics, and then they see this volcano plot, and then they found that that one of them. Mm-hmm. They don't specify, but probably they have all the data set included into the supplementary methods. And then just...
They didn't mention
really
that.
So they just say, okay, and Pkm2 was one of that. Yeah, and since it is kind of a bottleneck to switch where pyruvate goes, either to lactate or TCA, so they decided to check it, right?
Yes.
Yeah. Okay. No, I agree. That's a great point. So even if it is not the major hit, not the top hit, like one of the major hits, but not the top one.
Right? The Pkm.
Yeah. This means that this coloring means that there is okay, I see what I mean. So there were better hits, but they still went for Pkm2 because they thought it's more interesting because it's a bottleneck for the switch and how to steer the pyruvate either to lactate or to the TCA. That's a great point. Exactly. So that is the way how good research actually can initiate like really nice and compelling results. I agree. That's a great point. Yeah, I agree with that. And actually, what I like is this kind of representation in figure 3. So figure 3C, I think I didn't see it before in this way. They show just the pathway, and within the pathway, they show the change in a colored way. It's really nice. It's really clear. I really like this. Really nice. Yeah, and I generally, I personally find like this very round and very very well designed story. So yeah, really cool. Nice. Where did they publish, by the way?
National
Neuroscience. Cool. Yeah, great. I think it deserves at least Nature Neuroscience, I would say. Yeah, so it's really nice. Yeah. Cool. Any other opinions, maybe? Comments? Okay. If not, thank you, everyone. Again, was a really nice discussion and also nice story you found. And really great job by Xian. So next week we'll continue. So that will be the last one for this semester, and that will be I don't know who is going to present from the Polish side, but we can.
I am next in line, so
cool.
It's my turn.
We're looking forward to that. Yeah. Okay. Thanks everyone, and have a great day. Bye bye. Bye bye everyone.
Thank you.
bye.